Look around you, at the vibrant tapestry of life, from the smallest microbe to the towering redwood, from the simplest single-celled organism to the intricate complexity of the human mind. Life is abundant, resilient, and endlessly fascinating. But pause for a moment and consider the most fundamental question of all: where did it all begin? How did the inert chemicals of an early planet transform into the pulsating, evolving phenomenon we call life? This is the grand enigma, the ultimate origin story, etched into the very fabric of our world. Our story begins some 4.5 billion years ago, not with lush forests or teeming oceans, but with an Earth utterly alien to us. Imagine a molten world, still cooling from its violent formation, constantly bombarded by asteroids and comets. Volcanic eruptions spewed forth toxic gases, blanketing the planet. There was no blue sky, no breathable air. This was a planet in its raw, brutal infancy, a hostile cradle where the very concept of life seemed impossible. Yet, within this chaos, the seeds of existence would somehow be sown. As the planet slowly cooled, a thin crust began to form, but the atmosphere remained a suffocating, alien cocktail. It was dominated by gases like methane, ammonia, carbon dioxide, and copious amounts of water vapor, a far cry from the oxygen-rich air we breathe today. There was no ozone layer to shield the surface from intense ultraviolet radiation, and frequent, colossal meteorite impacts continued to reshape the young planet. This wasn't a world inviting to life, it was a cauldron of extreme conditions, pushing the boundaries of what was chemically possible. Crucially, for life as we know it, water had to arrive. Scientists believe much of Earth's water was delivered by icy comets and water-rich asteroids crashing into the surface over millions of years. Simultaneously, massive volcanic outgassing released vast quantities of water vapor from Earth's interior, which then condensed to form torrential rains. Over time, these rains filled the vast depressions in the planet's crust, giving birth to the first, searingly hot, highly mineralized oceans. The stage was set for chemistry to truly begin its dance. With oceans established, a revolutionary idea began to take shape in the minds of scientists like Alexander Oparin and J.B.S. Haldane in the 1920s. They proposed that these early oceans weren't just water, but a primordial soup, a vast chemical reactor. In this soup, simple inorganic molecules, energized by various forces, could react and combine to form more complex organic compounds. It was a bold hypothesis, suggesting that life didn't need a divine spark, but could arise from the very chemistry of the planet itself, given the right conditions. For inorganic molecules to transform into organic ones, energy was paramount. The early Earth provided an abundance of it. Intense ultraviolet radiation, unhindered by an ozone layer, constantly bathed the planet's surface. Frequent lightning storms ripped through the thick atmosphere, delivering powerful electrical discharges. Heat from relentless volcanic activity, both above and below the nascent oceans, also provided thermal energy. These powerful forces acted as the fires of creation, driving the otherwise improbable chemical reactions necessary for life's earliest building blocks. Under these energetic conditions, the simple inorganic molecules such as water, methane, ammonia, and carbon dioxide began to break apart and recombine in new ways. Over immense spans of time, this process led to the formation of more complex organic molecules, the fundamental building blocks of life. Chief among these were amino acids, the monomers that link together to form proteins, and nucleotides, the units that construct DNA and RNA. These were humble beginnings, but they represented a profound leap from inorganic chemistry towards biological complexity. In 1952, American scientists Stanley Miller and Harold Urey put the primordial soup hypothesis to the test with a groundbreaking experiment. They built an apparatus that simulated the conditions of early Earth, a flask of water representing the ocean, heated to create vapor, and another containing the presumed early atmosphere gases, methane, ammonia, hydrogen. Electrodes delivered electrical sparks to simulate lightning, and a condenser cooled the gases, causing rain to fall back into the ocean. It was an ingenious attempt to recreate abiogenesis in a bottle. The results of the Miller-Urey experiment were astonishing. After just 1 week, the ocean flask had turned murky brown. Analysis revealed that a variety of amino acids, the very building blocks of proteins, had been synthesized spontaneously within the apparatus. This landmark experiment provided compelling evidence that organic molecules, crucial for life, could indeed form from inorganic precursors under plausible early Earth conditions. It was a powerful validation of the primordial soup concept, demonstrating that the chemical origin of life was not merely theoretical, but experimentally demonstrable. While groundbreaking, the primordial soup hypothesis and Miller-Urey experiment also presented challenges. The intense UV radiation that provided energy could also rapidly destroy nascent organic molecules on the surface. Furthermore, the vastness of the early oceans meant significant dilution, making it difficult for molecules to concentrate sufficiently to react. And how would these simple monomers then link together, or polymerize, to form complex chains like proteins and nucleic acids in an aqueous environment? Water itself can break these bonds, presenting a chemical paradox. These challenges led scientists to consider alternative environments for life's genesis, shifting focus from the surface to the deep. In the late 1970s, the discovery of hydrothermal vents on the ocean floor opened up a new, exciting possibility. These deep-sea geysers spew superheated, mineral-rich water from Earth's interior, creating unique chemical and thermal gradients. Here, protected from destructive UV radiation and meteorite impacts, and with a constant supply of chemical energy, the conditions seemed far more conducive for complex organic chemistry to unfold. Among the various types of hydrothermal vents, alkaline vents have garnered particular interest. These vents are typically cooler and release water rich in hydrogen gas and methane, creating fine, porous mineral structures. The interface between the vent fluid and the acidic ocean water creates natural proton gradients, similar to those used by modern cells to generate energy. These tiny, mineral compartments could have acted as natural protocells, concentrating reactants and providing protected microenvironments for the earliest biochemical reactions, even before membranes evolved. Within the intricate structures of hydrothermal vents, certain minerals, particularly those involving iron and sulfur, are thought to have played a crucial role. These iron-sulfur clusters can act as catalysts, facilitating chemical reactions that would otherwise proceed very slowly or not at all. This provided a pathway for the synthesis of more complex organic molecules, including those that could form rudimentary metabolic pathways, the beginnings of how cells generate and use energy. These mineral catalysts offered an elegant solution to the problem of speeding up prebiotic chemistry. Beyond catalysis, hydrothermal vents offered vital protection. The deep ocean shielded nascent organic molecules from destructive ultraviolet radiation and the incessant bombardment of meteorites that still plagued the surface. The intricate, porous structures within the vents also served to concentrate dilute reactants, overcoming the dilution problem faced by the primordial soup. This unique combination of protection, energy, and concentration made alkaline hydrothermal vents a highly compelling candidate for the birthplace of life, providing a stable, dynamic environment over vast geological timescales. Whether in the primordial soup or deep-sea vents, forming simple organic molecules was only the first step. The next, far more challenging hurdle was polymerization, linking these smaller monomer units into long, complex polymer chains. Amino acids had to link to form proteins, and nucleotides had to join to create nucleic acids like RNA and DNA. This process often involves the removal of a water molecule, which is thermodynamically unfavorable in a watery environment. Overcoming this chemical barrier was critical for the evolution of true macromolecules. To address the chicken and egg problem, which came first, genes, DNA/RNA, or enzymes, proteins, the RNA world hypothesis emerged as a powerful solution. This theory proposes that RNA, not DNA, was the primary genetic material and the main catalyst of early life. RNA molecules, or ribozymes, could both store genetic information and catalyze biochemical reactions, fulfilling both roles currently performed by DNA and proteins respectively. It offers a simpler, more elegant pathway for the emergence of self-replicating, evolving systems. RNA's versatility makes it uniquely suited for this proposed ancient role. Unlike DNA, which is primarily a stable information storage molecule. RNA is single-stranded and more flexible, allowing it to fold into complex 3D shapes. These shapes enable it to act as an enzyme, or ribozyme, capable of catalyzing reactions like breaking and forming chemical bonds, and even catalyzing its own replication. This dual capability, storing information and performing work, is what makes the RNA world hypothesis so compelling as a bridge between inert chemistry and biological life. A critical step in the RNA world would have been the ability of RNA to replicate itself. While complex modern cellular machinery handles DNA replication with incredible precision, early RNA replication would have been far simpler, perhaps error-prone, but crucially, self-propagating. Some laboratory experiments have demonstrated that RNA molecules can act as templates for the synthesis of complementary RNA strands, even in the absence of protein enzymes. This primitive form of self-replication is the very essence of heredity, allowing successful molecular arrangements to be passed on and evolve. Even RNA, with its complex sugar-phosphate backbone and nitrogenous bases, might have been too sophisticated to emerge spontaneously. This has led to the exploration of pre-RNA world hypotheses, suggesting even simpler genetic polymers might have existed first. Molecules like peptide nucleic acid, PNA, or threose nucleic acid, TNA, are being investigated. These alternatives have simpler backbones yet can still store genetic information and base pair, potentially forming a stepping stone to the more complex RNA system. The search for the truly first genetic molecule continues. For chemical reactions to become truly lifelike, they needed a boundary, a way to separate the internal chemical environment from the chaotic external world. This crucial step was the formation of protocells. Imagine a simple membrane encapsulating a collection of self-replicating RNA molecules and the chemicals they needed. This membrane would allow the concentration of reactants, protect nascent genetic material from harsh external conditions, and create a distinct, controlled space where metabolism could begin to develop. It was the birth of individuality. The spontaneous formation of these early membranes is attributed to the unique properties of lipid molecules. Lipids are amphiphilic, meaning they have both water-attracting, hygrophilic, and water-repelling, hydrophobic, parts. When placed in water, these molecules spontaneously arrange themselves into bilayers, forming vesicles or sacs. This self-assembly is a natural, energetically favorable process, providing a plausible mechanism for the emergence of the first protocell membranes. This simple chemical property proved to be a profound enabler of life's cellular structure. These rudimentary protocells would have possessed several key advantages. By concentrating reactants within their boundaries, they increased the efficiency of chemical reactions, favoring the formation of longer polymers and more complex metabolic pathways. They also offered protection from external degradation and fluctuations. While not yet alive in the modern sense, these protocells represented a critical intermediate stage, demonstrating rudimentary forms of compartmentalization, localized chemistry, and perhaps even simple growth and division, laying the groundwork for true cellular life. The transition from a protocell, a non-living chemical system, to a true living cell is a blurry yet profound boundary. It marks the moment when a system acquires self-sustaining metabolism, reliable self-replication, and the capacity for evolution through natural selection. This wasn't a single eureka moment, but a gradual process of increasing complexity and integration. The genetic information became more stable, the metabolic pathways more efficient, and the cell membranes more sophisticated, eventually leading to the first entity we would unambiguously recognize as alive. While we may never identify the very first living cell, scientists have converged on the concept of LUCA, the last universal common ancestor. LUCA is not the first life form on Earth, but rather the single organism or population of organisms from which all current life on Earth descended. It represents a critical bottleneck in the tree of life, a point of shared heritage for every plant, animal, fungus, and bacterium alive today. LUCA is estimated to have lived between 3.5 and 3.8 billion years ago, after life had already diversified somewhat. By studying the fundamental characteristics shared by all modern life, we can infer much about LUCA. It possessed a DNA-based genome, used RNA for genetic information transfer, and had a sophisticated system for protein synthesis involving ribosomes. It utilized ATP as its primary energy currency and conducted basic metabolic reactions, including carbon fixation. These shared molecular mechanisms are the indelible signatures of our common lineage, powerful evidence for LUCA's existence and its profound impact on the subsequent evolution of all biological diversity. The concept of LUCA implies either a single, unique origin event for life, or more likely, that while life may have started multiple times, only one lineage ultimately survived and proliferated. All other early forms of life either died out or were outcompeted by LUCA's descendants. This evolutionary bottleneck ensures that we, and every other organism, carry the molecular echoes of this distant ancestor, a testament to its successful strategies for survival and reproduction in the challenging early Earth environment. It represents a single, victorious branch on the tree of life. While LUCA itself is a theoretical construct, direct evidence of early life exists in the geological record. Some of the oldest undisputed fossils are stromatolites, layered rock structures formed by microbial mats. These ancient formations, found in places like Western Australia, date back an astonishing 3.5 billion years. They represent the macroscopic evidence of microscopic life, built layer by communities of early microorganisms, offering a tangible link to life's earliest pioneers and their profound environmental impact. The organisms responsible for forming these early stromatolites were predominantly photosynthetic bacteria, such as cyanobacteria. Critically, these were the first organisms to evolve oxygenic photosynthesis, a process that released oxygen as a byproduct. Over immense spans of geological time, billions of years, this seemingly simple metabolic innovation slowly but inexorably transformed Earth's atmosphere. From an anoxic, toxic brew, our planet began its journey towards becoming the oxygen-rich world that enabled the evolution of complex, multicellular life, including ourselves. The accumulation of free oxygen, primarily due to photosynthetic microbes, culminated in what scientists call the Great Oxidation Event, or GOE, roughly 2.4 billion years ago. This wasn't a sudden burst but a protracted period of change that fundamentally altered Earth's geochemistry. While it led to a mass extinction for many anaerobic life forms, it also paved the way for the evolution of aerobic respiration, a much more efficient way to generate energy. This planetary-scale transformation was a prerequisite for the eventual emergence of eukaryotic cells and, much later, complex animal life. While abiogenesis on Earth remains the leading scientific explanation, another intriguing hypothesis offers a different perspective on life's origins: panspermia. This theory suggests that life's building blocks, or even simple microorganisms, could have been delivered to Earth from space, perhaps hitching a ride on meteorites or comets. It doesn't explain how life originally arose, but rather how it might have traveled between planets or even star systems, suggesting that life might be a cosmic phenomenon, seeded across the universe. Evidence supporting panspermia comes from the discovery of organic molecules in meteorites. The Murchison meteorite, which fell in Australia in 1969, famously contained a diverse array of amino acids, as well as nucleobases and sugars, many of which are identical to those found in terrestrial life. These extraterrestrial organics demonstrate that the raw materials for life can form readily in space and survive the harsh journey through our atmosphere, lending credence to the idea that Earth's primordial soup might have had some key ingredients delivered from beyond. It's important to clarify what panspermia does and doesn't explain. While it provides a fascinating mechanism for life's delivery to Earth, it doesn't solve the fundamental question of how life first originated. If life came from another planet, the question simply shifts to how did life start on that planet? Nevertheless, panspermia highlights the interconnectedness of our solar system and potentially beyond, suggesting that the ingredients for life, and perhaps even life itself, might be far more widespread in the cosmos than we once imagined. A more speculative variant of panspermia is directed panspermia, proposed by Francis Crick and Leslie Orgel. This intriguing hypothesis suggests that life on Earth was deliberately seeded by intelligent extraterrestrial beings, perhaps sending a spacecraft carrying microorganisms or even genetic information to our planet. While there is no empirical evidence to support this idea, it pushes the boundaries of our imagination, inviting us to consider whether our existence might be part of a grander, cosmic design, or an experiment by an ancient civilization. One of the profound mysteries in abiogenesis is the origin of biological homochirality. Organic molecules can exist in two mirror image forms, like a left and right hand, called enantiomers. Yet, all life on Earth exclusively uses L-amino acids to build proteins and D-sugars for nucleic acids. Why this preference? Random chemical synthesis produces an equal mix of both forms. How did life select and propagate only one-handedness so universally? This puzzle suggests a very early, critical selection event that set a fundamental constraint for all subsequent biology. Scientists propose several theories to explain biological homochirality. One idea involves mineral surfaces, or specific crystal structures might preferentially absorb or catalyze the formation of one enantiomer over the other. Another hypothesis points to circularly polarized light, which can exist in space or near certain active geological sites, selectively destroying one enantiomer. Or perhaps it was a purely random event, a chance selection that, once established in the first replicating systems, became fixed due to the advantages of molecular uniformity. The answer remains elusive, but vital. Another challenge for abiogenesis is understanding how incredibly complex systems, like the intricate machinery of a cell, could have arisen through gradual, undirected steps. This is often framed as the irreducible complexity argument by creationists. However, scientific models propose that such systems evolve from simpler, functional precursors, with each incremental addition conferring a selective advantage. What might appear irreducibly complex now was likely built piece by piece, with parts initially performing different functions, gradually co-opting and integrating into sophisticated molecular machines. While scientific principles drive the search for life's origins, the role of chance, or rather, probability, cannot be ignored. The vast number of potential chemical reactions, the sheer scale of geological time, and the alignment of specific environmental conditions suggest that many improbable events had to converge for life to emerge. It wasn't necessarily a unique cosmic lottery win, but a series of favorable probabilistic pathways that eventually led to a self-sustaining system. Life might be an expected outcome given enough time and the right physics, but the exact path taken could have been incredibly specific. The quest for life's origins is far from over. It is a vibrant, interdisciplinary field of active research. Scientists across the globe continue to conduct new laboratory experiments, simulating early Earth conditions with increasing sophistication. Deep-sea exploration continuously reveals new insights into hydrothermal vent ecosystems. Planetary science missions to Mars, Europa, and Enceladus search for signs of past or present extraterrestrial life, offering comparative insights into the conditions that might favor abiogenesis. Every discovery adds another piece to this grand, cosmic puzzle. The search for life's origins on Earth is deeply intertwined with the burgeoning field of astrobiology, the study of life in the universe. If life could arise here, could it have arisen elsewhere? The discovery of exoplanets in habitable zones, some with evidence of liquid water, fuels this inquiry. Missions to icy moons like Europa and Enceladus, with their subsurface oceans and hydrothermal activity, represent prime targets for finding independent origins of life. Understanding abiogenesis on Earth provides a crucial framework for recognizing potential life signatures across the cosmos. Before we can fully understand how life began, we must grapple with a seemingly simple yet profoundly complex question: what is life? There's no single, universally accepted definition. Biologists often point to a set of characteristics: organization, metabolism, growth, response to stimuli, reproduction, and evolution. Yet, a crystal grows, a fire metabolizes, and a robot can replicate. The boundary between complex chemistry and simple biology is fuzzy, making the study of abiogenesis not just about discovery, but also about refining our very understanding of existence itself. Even in our modern, DNA and protein-dominated world, RNA retains critical roles, serving as a constant reminder of its ancient, central position. Messenger RNA, mRNA, carries genetic instructions from DNA to ribosomes, where transfer RNA, tRNA, helps assemble proteins. Ribosomal RNA, rRNA, forms the structural and catalytic core of ribosomes themselves. Beyond protein synthesis, numerous non-coding RNAs regulate gene expression. Viruses, such as influenza and HIV, still utilize RNA as their primary genetic material, showcasing the molecule's fundamental importance and versatility across all forms of life. At its core, life is a thermodynamic process, a system that maintains order by exploiting energy gradients. Whether from sunlight, lightning, or chemical differences at hydrothermal vents, early life harnessed these energy flows to drive otherwise unfavorable reactions. The ability to capture and convert energy, often through redox reactions, was fundamental for building complex molecules, sustaining nascent metabolic cycles, and eventually powering cellular functions. Life isn't just matter, it's matter organized and energized in a perpetual dance against entropy. The origin of life was not a singular, instantaneous event, but rather a long, continuous process, a gradual ascent from geochemistry to biochemistry, from non-living matter to self-replicating systems. Imagine a continuum, where the boundaries between stages are fluid. Simple organic molecules accumulated, polymerized into complex chains, became encapsulated in membranes, developed rudimentary self-replication, and began to evolve. Each step, however small, built upon the last, transforming the inert into the living over hundreds of millions of years, an unfolding drama on a planetary scale. Earth's geological processes played a crucial, often overlooked, role in setting the stage for life. The deep carbon cycle, driven by plate tectonics and volcanic activity, recycled essential elements from the planet's interior to its surface and atmosphere. Volcanic outgassing released carbon dioxide, a key ingredient for organic molecules and atmospheric chemistry. Subduction zones and crustal recycling ensured a constant renewal of geological environments, providing fresh mineral surfaces and chemical gradients critical for prebiotic chemistry to persist and evolve over eons. The lack of free oxygen in the early Earth's atmosphere was not merely a challenge but a necessity for abiogenesis. Oxygen is highly reactive, it would have rapidly oxidized and destroyed the delicate organic molecules forming in the primordial soup or at hydrothermal vents, preventing their accumulation and polymerization. The anoxic conditions created a reducing environment, conducive to the synthesis and stability of complex organic compounds. Only after life had firmly established itself and evolved sophisticated ways to protect itself from oxygen, could the atmosphere begin to transform. Even before the Great Oxidation Event, early life forms were harnessing energy from light. The earliest forms of photosynthesis were likely anoxygenic, meaning they used light energy to convert carbon dioxide into organic matter without producing oxygen as a byproduct. These primitive light harvesters, perhaps using different pigments than modern plants, represented a significant step in gaining metabolic independence. They demonstrated life's capacity to tap into an abundant, renewable energy source, setting the stage for the later evolution of oxygenic photosynthesis and its profound planetary impact. The emergence of life wasn't just about individual chemical reactions, it was about the intricate interplay of Earth's vast geochemical cycles. The circulation of water, the weathering of rocks, the volcanic outgassing, and the cycling of elements like carbon, nitrogen, and phosphorus all created a dynamic, evolving environment. These cycles provided the raw materials, the energy gradients, and the physical spaces necessary for prebiotic chemistry to operate. Life didn't just appear on Earth, it emerged from the very fabric of Earth's interconnected geological and atmospheric systems. Beyond their catalytic roles, mineral surfaces offered additional advantages for early life chemistry. Clay minerals, for example, have charged surfaces that can attract and concentrate organic molecules, overcoming the dilution problem. They can also protect these molecules from UV degradation and help stabilize long polymer chains, potentially acting as templates for their assembly. Certain minerals like pyrite might even have provided both a surface for attachment and a source of chemical energy through their oxidation, creating dynamic microenvironments where protocells could thrive. One of the most profound chicken and egg problems in abiogenesis is the origin of the genetic code itself. DNA, and RNA, carries instructions for building proteins, but proteins are required to read those instructions and build more proteins and nucleic acids. How could this complex, interdependent system arise simultaneously? The genetic code is universal, suggesting a common origin, yet its arbitrary nature, which codon codes for which amino acid, remains a mystery. Scientists hypothesize that the earliest code was simpler, perhaps encoding fewer amino acids, gradually evolving to its current complexity. Despite incredible progress, many gaps in our understanding of life's origin remain. We lack a complete, step-by-step pathway from non-living chemicals to the first living cell, and many proposed stages still require more experimental validation. Alternative theories, sometimes quite radical, continue to be explored. This isn't a failure of science, but a testament to the immense complexity and unique nature of the problem. It highlights the vast unknown that still surrounds us, fueling curiosity and driving scientists to push the boundaries of our knowledge. Beyond the scientific specifics, there is a profound beauty in the story of abiogenesis. It's the story of intricate order emerging from seeming chaos, of simple elements self-organizing into astonishing complexity. It's a testament to the inherent creativity of the universe, where the laws of physics and chemistry, given enough time and the right conditions, can spontaneously generate something as magnificent as life. This process, spanning billions of years, is one of the most elegant and awe-inspiring narratives in all of science, reminding us of the wonder of existence itself. Each of us, every living being on Earth, carries within ourselves the molecular echoes of this ancient history. Our DNA, our metabolic pathways, the very mechanisms that define us as alive, are direct descendants of those first, fragile chemical systems that emerged billions of years ago. We are a continuous, unbroken lineage stretching back to Luca, and perhaps even further to the very first protocell. This understanding connects us not only to all other life on Earth, but to the fundamental processes of the universe, making us truly children of the stars and the primordial Earth. The question of how life began on Earth remains one of humanity's most profound and enduring quests. It challenges our assumptions, pushes the boundaries of scientific inquiry, and forces us to reconsider our place in the cosmos. While we have made remarkable strides in uncovering the plausible pathways, the complete picture is still emerging. But it is in this ongoing pursuit, this relentless curiosity, that we not only seek to understand our past, but also to glimpse the potential for life's emergence elsewhere, making us eternal explorers of life's greatest secret.